Okay, welcome to the Botanic Gardens Biosecurity webinar series. I'm Daniela Carnfale, I'm from Plant Health Australia, and I'm one of the project officers as part of the project team. A little bit about these webinar series. The webinar series is being coordinated by the Botanic Gardens Biosecurity Network. The network is coordinated by Plant Health Australia with support from expert collaborators. And it's funded by the Australian Government's Agricultural Competitiveness White Paper, the government's plan for a stronger farms and a stronger economy. So just before we get into today's webinar, I just want to go through a few housekeeping things with all of our online attendees. So today we have a panel, and the panel is comprised of the project team, and they are going to assist me running the webinar. They'll also be assisting, looking at answering any questions that you may have during the webinar. So today, what we would like you to do is, if you have any questions throughout the webinar, if you could use the chat box to ask your questions, you can ask it to individual panelists or to everyone, and you can do that throughout the webinar. Hopefully what we'll do is at the end, we'll have a little bit of time and I will take some of those questions and relay those onto Peter today so that he can answer them. Any questions that we don't get a chance to answer, we will take those on and provide a written response to all attendees. This webinar will be recorded and it will be available at our Botanic Gardens Biosecurity Network site, as well as through our Facebook page. Now, I'd just like to pass on over to Peter Symes, who will be presenting today's webinar, and he'll be looking at surveillance and partnerships for plant biosecurity in the Melbourne Gardens.
Okay, hello everybody. Thank you for coming to hear this presentation. And usually I make a joke at this point about I'll be asking questions at the end and asking questions of the audience as a bit of a test, but I can't do that, so we'll have to move through it. But hopefully you find this interesting. So just to provide some context for Melbourne Gardens, so Royal Botanic Gardens Victoria is 2 sites. We have also the Cranbourne Gardens, but I'm talking about the Melbourne Gardens today. And I don't know if you can see the mouse here, but the Botanic Gardens is this sort of bit of the green, lovely green area that you see, this bit around here. And we're comprised of 39 hectares of land. The rest of that green space that you see is 67 hectares of the domain parklands which surrounds the gardens. And that presents a risk in itself for us because of pest disease that might occur in those parklands, being able to jump across into our site. But there are other risks as well in terms of our context. We have international entries into Melbourne, and with COVID-19, a lot of this has dropped off, but we do have increasing globalisation and transport and entry of internationals into Australia previously. So with the port, ships can come in, they can have pests and diseases basically hitchhiking 5 kilometres away from the Botanic Gardens. The international airport over here, 20 kilometres away, international visitors can arrive, jump in a taxi, they might have dirt on their shoes, they can walk straight into the gardens with some new disease. The other risks that we have to face are, they've got the central business district not that far away, people can move in and out from there. We have a sports precinct which often has international athletes but also visitors, and it's just right over the bridge from the gardens. And we can't forget other issues such as tourist attractions like the Shrine of Remembrance. Don't know if you can see this here, but a lot of people come and visit that. But we can't exclude residential gardens, and they're right next door, they're part of our catchment. If a new exotic pest or disease arrives in that area, they're right next door, so things can move straight into the gardens. So because of all this, the Melbourne Gardens is considered a high-risk pathway because of its plant diversity and its proximity to all these vectors, if you like. I just wanted to share this image with you. I took this in Singapore, and I think it is a plant health professional's delight to see a lovely fence and a very strict sign saying, "Don't come in." But in the Botanic Gardens, of course, we can't have this fence around the outside and this sort of signage, though some of us at times wish we could, but we can't do that. And so for us at Melbourne Gardens, we've got limited control of access. The Melbourne Gardens is closed at night and locked up, but during the day, people can walk in freely. And unfortunately, some people think that it's okay to walk onto garden beds, which is sort of against our rules, but you know, you've got to get that selfie or the latest Instagram picture, and they will walk onto a garden bed to do that, and some spots get very heavily trafficked. Of course, if they've got soil on their feet, spores, they're walking those diseases straight into our gardens. But there are other issues and challenges facing us. Firstly, we've got a really high plant diversity in Melbourne Gardens. We've got 8,000 taxa or different kinds of plants, and the distributions of those taxa represent 98% of the world's countries. So we've got a lot of different plants, a lot of potential for different pests and diseases to host on our site. We have climate change, which is basically a moving goalpost. So temperature and rainfall, environmental conditions are changing. New pests and diseases can be advantaged by that. Some might be disadvantaged and some will drop out. And who knows what's next around the corner with this change. And then we have new pests, and myrtle rust appeared in Australia in 2010. And that's an image of myrtle rust there on one of our Lophamurtus, a New Zealand species. But some of these new pests could change hosts. So it might be new pests, but it might, with the changing conditions, suddenly appear on new plants. Just want to quickly cover off that the Royal Botanic Gardens Victoria does actually have a biosecurity policy and procedures to manage our biosecurity of our living collections and landscape. I'll come back to the principles. I just want to talk about the border controls and pest triangle schematic that you see there. So that's focus of two areas that we work on. One is how do we manage incoming and outgoing materials, landscape materials and plants? So how do we prevent incursions of exotic pests? And how do we prevent excursions of exotic pests? The Botanic Gardens does not want to be responsible for damaging industry or other gardens and landscapes. So what can we do to manage that boundary? One of the ways that we can do that is we close the loop, and we generally pretty much generate all our own compost. So all our green organics, all the material that we put on our garden beds is generated in-house. We don't have to bring any in. So we avoid the inadvertent risk of bringing something in to the gardens. Or if we do have to order soil, which we try to limit, what quality assurance can we put on that soil to ensure we don't bring a new pest or disease into the gardens? The other side of it is the infamous pest triangle, and some of you listening will know that very well. But essentially, it's a concept to denote whether you're at risk from a disease. So, for example, if you have the host plant, a susceptible host plant, you have the environment that will support that host, and you have the pathogen present, then there's a risk that you'll have disease. You take one of those factors out of the equation, then it's less likely or probably won't have any disease at all. And integrated pest management, so being more holistic about it, really rests on this foundation, and this is what we're doing within the gardens. So today we're going to focus on surveillance and pest plant sentinel programs, and networking and developing partnerships are the principles there. I want to show you a couple of case studies to show you what the risk is like, and as examples of surveillance. The next two, or the next three, I'm going to show you actually surveillance by horticulturalists in the gardens. We had this Phytophthora arrive in the gardens in 2000. Phytophthora asparagi, as it is known now. It was unknown then, it wasn't named. And you can see the sort of damage that it's causing to that Yucca gloriosa. That damage is actually off the ground, so it's infecting the tissues away from the soil level. And the agave attenuator that you see on your, hopefully it's your right, you can see the rotting and necrosis of the tissues there. So quite a severe, severely damaging pathogen. So when it arrived in September 2000, we lost 70 taxa, or 70 different types of plants within the family Asparagaceae. And some of the main ones for us are agave, Phocrea and Yucca. And any gardener lovers who are listening to this will know those plants well. But importantly, we also lost 10 wild collected, unique agave species that will probably never get into the gardens again. So out of our living collections, we lost 10 taxa that are probably close to impossible to have. So it's pretty devastating for us in the gardens. And at the time, this was considered to be a world first record on agave species. And you know that, probably recall my title was about when there's another world first. Well, we've got a few of them to show you. So really devastating on agave. Later on, we found it damaging bulbine vagans, which is in a different family, Asphodeliaceae, but in the same plant order of Asparagales, so in that Asparagaceae family. It's actually a native plant to New South Wales and Queensland, so you could imagine if this disease damaged that plant outside. One thing I'll leave with you on this case study was that this Phytophthora was also identified in Cranbourne from the vegetable growing areas where they're growing actually asparagus that you eat. And I sort of surmised whether, surmised that maybe the soils that were commonly collected from Cranbourne, the grey sandy loams, maybe we received delivery of that soil and brought this pathogen into the gardens. We don't know. The other one I want to share with you is this Helleborus necrosis virus. Appeared in the gardens. This appeared in October 2010. It was the first known record in Australia. We reported this to the appropriate authorities. They did surveys, and they actually found it in private gardens not that far out of Melbourne, including in asymptomatic plants. So not always visible, and essentially causes this stunting, this deformation, this sort of black netting and mottling. It really inhibits the vigour of the plants, but some plants don't always show the symptoms. It's believed to be transmitted by viruses, but that hasn't been proven that I'm aware of. So it is a difficult one to manage. And when we looked at where this disease occurred, the epicentre of these disease halos, if you like, were plants that were donated from one nursery. And I'll leave you to draw your own conclusions there. The other case study, and I'm moving more into partnerships now, on the 6th of March, we had a very severe hailstorm that hit Melbourne. Some of you might remember how bad that was. I was in an office preparing for a training week in Singapore on horticulture, and I had these large hailstones rolling into the doorway, and it basically shredded the gardens. You can see from the images the leaves on the path, the shredded leaves. The Guilfoyle's volcano project had just been completed, and some of you will be familiar with that. And a lot of the cacti up there were basically just shredded. This really set the scene for a new disease to hit the gardens. So later in June, horticulturists reported these unusual symptoms in pinus in the gardens, and we of course reported this to our friends in Agricultural Victoria, who very promptly came in and took samples, and then they discovered this new disease, and it was actually a first record in the world on pinus, Diplodia africana, and this had only been detected in prunus in Africa before this. We had to remove these trees because the risk was unknown, but there was a precautionary sort of approach taken, and we had to explore very different ways of trying to contain the material. Use plastic, it's slippery. If using a chainsaw, that brings other risks to it. So we trialled the use of weed mat, and that seemed to work reasonably well. And there's one of our staff there suited up and chopping down our Pinus muricata. Pinus muricata is actually considered rare in California and endangered in Mexico, and it was the only specimen in the gardens that we had. So we lost that tree. We also removed the Pinus patula. So eventually we removed two Pinus species under hygienic and sanitation. And you can see that image there, the sort of control zone set up to avoid cross-contamination. Everything had to be wrapped up. And there's David Smith there on the left from Agriculture Victoria, has been an absolute champion for us in the gardens. This disease has since been detected in junipers, but also Grevillea robusta in the Mediterranean sort of climates of the world. So it's not only an issue for pines. The pathogenicity testing for this disease basically concluded that it wasn't any worse than any other diplodias that occur in plantations. These diseases often need, I suppose, a previous stressful environment, and we had the millennium drought, and often damage to the trees, the hailstorm, which provided entry for this disease to cause this damage. Moving more directly into partnerships, one of the programs we've been working with the Chief Plant Health Officer, Agriculture Victoria, is these, I suppose, passive sort of surveillance since 2012, using pheromone traps, so essentially a trap that uses smell, if you like, to attract various insects on these panel traps, and they're the witch's hat-liking device that you might see there on the, hopefully your right. Basically, insects attracted to that trap, they hit the thing and then they fall down into a bucket and then they're collected and identified. But this acts as because of our high risk pathway, this acts as an early warning system, not only for us in the Botanic Gardens, but for Victoria more widely. So this is quite a responsible responsibility that we have to take. Nothing's been detected of any significance, but essentially we're looking for that host range there, pine soy bottle, beetles, Asian longhorn beetle, Asian gypsy moth, etc. Asian gypsy moth is one that's particularly good at hitchhiking on things like shipping containers. So you can see the correlation between the ports being nearby. The other project that we're working with the Chief Plant Health Officer Unit, AgVic, is screening catchments. And you can see in the image of that pink area, that's the Royal Botanic Gardens Victoria catchment. So we can actually scan that for Phytopthora. And the yellow area is adjacent catchments that we actually harvest stormwater. We're collecting stormwater to irrigate the landscape, about 70 megalitres a year on average, which basically significantly offsets our potable water use. At the same time, we're potentially bringing in risks. So we need to be sure the water we're collecting is not carrying potentially nasty pests or diseases. In California, particularly, they're doing waterway baiting for Phytopthora, which I'm going to talk about now, basically scanning for Phytopthora morum, or sudden oak death, a very significant disease for us here. A lot of people think it's only oats, but it's been found by various studies that this Phytopthora can actually impact quite a lot of native species as well. If it lands in that yellow area, we've got a mechanism to detect it. Typically, there's plant material used and water collected to act as bait, so attractant for the zoospores. What's been typically used are leaves like rhododendron leaves, Eucalyptus cyberi or silvertop ash cotyledons, you can see there. But more recently, we've been using a vulnerable plant. So this plant's vulnerable in Victoria, but growing in the gardens. This Pimelia Trayvordii leaves. Because they're small, we can use quite a large number of leaves without damaging the plant. When I collected the sample in this bottle that you see, the leaves were all floating to the top and not wetting up at all. And I thought, oh, this is not going to work. How are the spores going to get into these leaves if they're basically sitting on the surface of the water? But they did actually work. And we'll come to that. So you can see from the image there, there's our fern gully here. So we're collecting water right down the bottom of the catchment. We're scanning this whole area here and all our catchment. You can see there's some sporangia of Phytophthora and the spores coming out. And those spores are attracted to the plant material in the water and swim to it and basically infect it. But also pear baits are used, so solutions and pears are used to attract Phytophthora. And then that material is then refined on agar that's specially designed for Phytophthora species. Now, Phytophthora are not a fungus. They're in oomyces. We call them fungus-like organisms, but they're not strictly a fungus as such. That DNA is sequenced, and these are the results that we've received so far. The asterisked Phytophthora, which were new and hadn't been detected in this method, were all retrieved from the Pimelia. So the not being able to wet the leaves didn't really seem to stop some of the zoospores swimming through the water. But these are the results that we obtained. Another partnership we've been sort of supporting on and off. It's been active, more active at times than others, but it's what's called the Better Biosecurity Program. And Better Biosecurity is basically a multi-party science sort of consortium looking at ways to better protect New Zealand's living assets from new exotic pests and diseases. But this program is really looking at the use of New Zealand flora in overseas destinations as an analogue, if you like, for what pests and diseases might affect New Zealand. And our New Zealand living collection, which you see an image of there, can act as a sentinel for our colleagues in New Zealand. If a pest or disease arrives on our site, it's a way of pre-warning them of potential risks to their flora. And there's been specimens collected from our landscape, taken across to New Zealand under phytosanitary conditions and checked for, I suppose, risk for their flora. The communication continues with this project. As new pests on flora identified, we've had myrtle rust on our Lophomertus plants. And interestingly, there's Lophomertus in the gardens that haven't been infected with myrtle rust, even ones growing right next to those that have been infected. So that's an interesting observation. It may be that there's genetic variation. There's might be even some resistance in the population. But when we had our first myrtle rust incursion in the gardens, we're able to report on this straight away to bee-free personnel, and they were very interested in our findings. The other partnership they're working with is a citizen science project, and it's supporting the Botanic Gardens Biosecurity Network. And this is being supported by the Melbourne Friends. Now, this was all well and good, and we're just starting this program when COVID-19 made its entry into the garden. So at the moment, this program's postponed, but I just want to talk a bit about how we're engaging with our volunteers and people interested in the gardens in a way of increasing the surveillance network, if you like. The people who sign up to this program, they basically have an app on their Android or Apple phone, and they can go out predetermined sites, take an image, write a report and send that in, and that's centralised information on whether they're detecting a disease or not. That all sounds lovely, lovely and simple and easy, but there's actually a bit of work to it. Firstly, some of these people don't necessarily know their plants very well, so there's training involved in plant identification, and it meant that we had to take a whole series of photographs of the sentinel plants to assist our interested volunteers, which I think was about 6 at the time, in doing this surveillance. And we also had to sort of come up with a map and break it up into areas that they could then tackle as their zone of responsibility, and try to have a replicate of the suppose exotic pests that we're looking for, so brown marmorated stink bug bug, myrtle rust, polyphagia shot-hole borer, rose rosette virus, which is not on the program, but one we wanted to check for, and stigmina leaf spot. And I'd like to thank David Gale, who's listening, who helped me actually set this up. But this is on one of the gardens maps. And this is what it looks like in terms of some of the plants. So we're not only looking at foliage symptoms and signs on foliage, we're also looking at bark and trunks for things like polyphagia shot-hole borer, which makes little holes. So there's a range of different hosts that the people conducting the surveillance have to look at. This program is not operating currently, but we're still doing bits of it and reporting out. So we've got these partnerships and surveillance that needs to be supported by records management. In the Melbourne Gardens, we actually have what we call a Pest module, which comprises part of our living collections database, which basically records every plant in the gardens. We're able to link pests and diseases that are diagnosed to that database to distinct plants or areas. And I'm just showing you on the right there, and we talked about world first. Here's probably another one, Fusarium falciforme. Now this is an interesting fungus because it's also can be a pathogen in humans and in animals. So it jumps across kingdoms. Probably in humans, it's mostly in people who have their immune systems compromised, but it's one that can jump across a number of life forms, if you like. I don't think it's necessarily a risk for us to be concerned about, but it's just interesting. So you can see the photograph there on the right, the rotting and damage at the base of the leaves. These plants actually recovered afterwards, the Phacroia quichiensis. This is the level of detail we can record. It acts as a very useful guide for horticulturalists who see these symptoms and think, oh, what could that be? Oh, it might be this. I better get that checked out. So it acts as a resource. And with the records to date in this database, that's what we're sitting at. So you can see most of the problems that are reported and that we get tested sitting, generally around fungus and oomyces or Phytoclean and Pythium. They're generally our biggest problems in the gardens that we have to deal with. And what does this look like? If you can map the occurrences, this doesn't actually show all the Phytophthora that we have. We've got quite a good range. We have about 20 species all up. But there's a way of managing your pests and diseases. If we know where they occur, we know what the topography of that land is like, we can make predictions about where certain Phytophthora might spread. It acts as a very useful visual tool for our horticulturists and curators in the gardens in terms of areas that they're working in. We have Phytoclean for washing down tools, so they know that they need to clean between these areas. So we can pretty much map any of these diseases in this database and produce a visual tool for managing these. For those who don't know, and I meant to mention this earlier, but Phytophthora literally means plant destroyer. So you can see why we're concerned about it. And I'm about to finish up, but just in summary, the partnerships are really important, and I really would like to give a really large thanks to Agriculture Victoria and their Chief Plant Health Officer unit, being incredibly supportive of us in the gardens. I've almost got a direct line. If we observe unusual symptoms, often the next day there's an officer out to check it out because they actually trust that we are indeed reporting something unusual. So we have an extremely strong relationship, and it's made a huge difference to our capability in biosecurity. And it is about partnerships and people working together. This image is us working on the first myrtle rust incursion that we had to face. And, it was a difficult thing, but it was fantastic to have that expert support. I would also like to thank Plant Health Australia for setting up the Botanic Gardens Biosecurity Network and really developing a much wider pool of expertise so that we can help each other. but ultimately all of us have a responsibility to watch out for a disease or pest problems, because it affects our enjoyment of our plants and landscapes. It also can affect our livelihoods. So all of us have a responsibility to be partners with one another in this area. Just in finishing up, I just want to provide further information and contacts if you're interested. These are some of the resources that are available to you. I'm happy for people to send me an email if I haven't covered some of the matter in adequate detail. The Botanic Gardens Biosecurity Network you probably know about, but the national priority list pests are national lists in Australia of the top priority exotic pests and diseases that we're concerned about, and it's a great learning tool to have a look at those. And there's a couple of papers there relating to some of the work that I've presented today. That's the end of my presentation. Thank you very much for listening.
Thank you, Peter. What a fantastic presentation. And I think we're really getting to understand how important it is to be looking out for pests and diseases in Botanic Gardens, and the pivotal role that Botanic Gardens play within that surveillance network, and also working in partnerships with multiple different organisations. So thank you again. What we'll do now is we'll get anyone to pop any questions into the chat box. We've had a few come in. We don't have a lot of time. We might just ask 1 or 2 questions today. So I'll just get straight into it. So could Diplodia africana affect native calitris?
That's a good question. I don't know the answer, but it's possible because it is jumping across families, so it could be a risk. I mean, it's been found in Grevillea robusta, so that's Proteaceae, very different family to pines. But I don't know. I'm not an expert. I can't give you a definitive answer on that one.
Yeah. Another question we've got come through is any of the identified Phytophthora native to Australia?
Another curly question for me. I don't believe so. Certainly the more severe ones aren't. Phytophthora cinnamomi is definitely not known to be native. But I would be relying on my biosecurity friends to be definite about that. Look, I think most of them are exotic, just because of the way that native plants respond to them, either in being damaged or impacted in some way.
Okay, fantastic. We might leave it there because we've run out of time today. Thanks again, Peter.
No
If you have any quick questions, you can pop them through and we can try and answer them offline and provide a written response. So you'll still have a little bit more time to pop any questions in. Just before everyone leaves, it'd be great if you can just take a moment to quickly fill out a poll for us, which should hopefully have popped up on your screen now. Once again, thanks, Peter, for your fantastic presentation. It was great.
Thank you. You're very welcome.
Now, whilst we're just waiting for participants to fill out that poll, we'll have a quick look at the webinar schedule coming up. So in 2 weeks' time, we've got a webinar from the Peri-Urban Environmental Biosecurity Network. So they're another community of practice, very much like the Botanic Gardens Biosecurity Network. And they're going to be talking a little bit about watching out for environmental pests.